Effects of potentially toxic elements on soil microbial composition. Potentially toxic elements, when accumulated in soil beyond their permissible limit, they firstly influence the quality and quantity of soil bacteria, fungi, actinomycetes, and other microbial population. Potentially toxic element contamination in soil not only produces different microbial community patterns but also change the chemical and biological properties of the soil. In the soils which are polluted by potentially toxic elements for a long time, those soil microorganisms which can specifically be adapted exist. The efficiency of microbial populations and organic mineralization is inversely correlated with the soil organic carbon content, an indicator of the impact of potentially toxic element pollution. Microbial communities in soils are very important as they are helpful in nutrient cycling, plant symbiosis, and the detoxification of noxious chemicals, used to control plant pests and plant growth. When metal-enriched sewage sludge is added to soil's microbial biomass, leads to a decrease in functional diversity and changes in microbial community structure. However, metal exposure may also lead to the development of metal-tolerant microbial population. Potentially toxic elements affect the microbial activity and microbial community structure. The number of bacteria and actinomycetes significantly decreased as compared to the control, while no significant difference in fungal cells up to 2 weeks of incubation of potentially toxic elements. The negative effect of studied potentially toxic elements on culturable heterotrophic bacteria was more than actinomycetes and fungi. The DGGE profile indicated that the bacterial community structure was changed in the CD/PB amended samples, particularly at high concentrations, but not bacterial taxon richness and community composition. The relative abundance of specific bacterial taxa including acidobacteria, actinobacteria, bacteroidetes, chloroflexi, planktomycetes, and probacteria is affected by potentially toxic elements pollution. A significant correlation between a group of metal resistance genes and metal concentration in soil was also reported by Azerbad et al. Acidobacteria GP and Proteobacteria thiobacillus bacteria had more links between nodes and more positive interactions among microbes in Cl and Ch networks were positively correlated with cadmium concentration, while Longilinea, GP2 and GP4, had fewer network links and more negative interactions in Cl and Ch networks were negatively correlated with CD. There was the only positive correlation in between the members of the phyla Creenarchaeota and Euryarchaeota, class Thermoproti and order Thermoplasmatales in CD metal, and had more network interactions in Ch networks. Lee et al. Also reported that, the microbial community composition, as well as a network interaction, was the shift to strengthen adaptability of microorganisms to potentially toxic element contamination. 2. Archaea was resistant to potentially toxic element contamination and may contribute to the adaptation to potentially toxic elements. PB2+ in soil showed a weak impact on the diversity of soil bacteria community, but it influenced the abundance of some genera of bacteria, as well as soil physicochemical properties. At the genus level, there was a significant difference in the relative abundances of heavy metal resistant bacteria such as Bacillus, Streptococcus, and Arthrobacter. The abundance of main bacteria phyla was highly correlated with total PB. The relative abundance of gymnotiminates, nitrospirae, and planktomycetes was negatively correlated with total PB. Lead influences both the microbial community composition and physicochemical properties of soil.